Today we'll be presenting on using machine learning to find trends in what constitutes as a healthy or unhealthy food. I'm Serena Chen.
I'm Lahari Karmuchi.
And I'm Atharva Patel.
Food is a very important part of everyday life. It provides us with nutrients. However, as with all things, there are disadvantages along with benefits. Many processed or artificial foods can harm people's health, leading to increased heart problems, obesity, and more. Thus, we adapted to create a program that could analyze whether foods were healthy or not. By using machine learning models in Python to see trends in healthy foods and unhealthy ones, compiling a dataset consisting of 17 different types of nutrients, and labeling them as healthy or unhealthy, we could run machine learning algorithms to determine their healthiness. Before we began, we made predictions regarding the results. We predicted that meats would be unhealthy due to high levels of saturated fats. Due to the amount of fibers, vitamins, and minerals in fruits and vegetables, we said they would be healthy. We also assumed that foods with high amounts of sugar and sodium would be unhealthy. Many factors can affect the healthiness of foods, such as carbs, lipids, sodium, and more. For a food to be healthy, it needs to have at least 0.8 grams of protein per kilogram of body weight. Carbohydrates should also make up 45% to 65% of daily calories. Lipids should be between 300 and 700 calories. Men should consume 150 calories of food a day, and women 100. From there, we branched out into other nutrients, such as calcium, iron, sodium, cholesterol, and more. And now for our process.
So first step of the process is data collection. For data collection, we had to find online. So we basically found data from the Food Data Central website, which was created by the US Department of Agriculture. Using this data, we retrieved lists of nutrients, which are categorized by food names. So in order to do this, we had to use a series of steps. The first step is to download information as .csv files. The next step is to use the FTC_ID in order to cross-reference different Excel files and organize correctly. The last step is to compare the FTC_IDs and nutrient IDs in order to compile a single Excel file that includes the food IDs, names, and various nutrients of each food in the Food Data Central database for our data collection. The next step is data labeling. So in order to do data labeling, we had to use human intuition in order to label food. We had to determine if foods were healthy or not by using two columns, which is the yes column and the no column. One of the columns is used to determine whether foods are healthy or not, while the other column provides the reason why, like the reason why we think the food is unhealthy or the reason why I think the food is healthy. So data is then imputed manually based on common knowledge. So just depending on whether we think it's healthy or unhealthy. So for example, if the food was a fruit or vegetable, we'll label it as healthy. Most meats are determined as unhealthy, and if the sugar content is too high, that'll be determined as unhealthy as well. The last step of the process is data cleaning. For data cleaning, we have two primary approaches in order to clean the data. The first step is to drop all rows with NaN and just create a model on the data left. The problem with this is that we have too little data left, and that causes the model to have too little training data, which is bad for us. The second primary approach is to use KNN imputer in order to impute all the missing values. The problem with this is that it caused a lot of overfitting since many of the values were too similar to each other, which would then lead to a bad model, which is not good. So we then decided to compromise these two models by dropping over the rows with two or more NaN values and then imputing the rest of the values.
To gain a better understanding of our data, we graphed all the nutrients in a bar graph to visualize their distributions. So on the left, you can see a bar graph of carbohydrate frequency and its distribution throughout the data. And on the right is the same thing with water. Correlation. Another important thing to look at was the correlation between all the nutrients to see the relationships between the specific nutrients and any other one. The most important one is the row on the bottom, and although it's really hard to read, it says healthy and it correlates it with every single nutrient. And basically that bottom row tells us the correlation of all the nutrients with whether the food is healthy or not. The highest correlation is between healthy and energy, with the correlation at -42%. And on the positive end of the spectrum, only water content stands out at about 32%. A few other interesting values include a -92% correlation between energy and water, as well as a 75% association between lipids and energy.
Now for our debrief.
Results. The results that we found were very interesting. The primary factors that we assessed were feature importance, confusion matrix, and accuracy score. Our best accuracy score was 90%. We used a confusion matrix to get a better understanding of how accurate our model really is. And after randomly selecting a random sample of 111 rows 6 times, we got about 37% of our data as a true negative, meaning that it was labeled healthy, but the model guessed unhealthy, and 15% false positive, meaning that it was labeled unhealthy, but the model guessed healthy. And finally, we used feature importance to figure out which value our classifier was depending the most to make its classification. This is the most important result of our experiment and tells us which nutrients we should assess the most when determining whether a food is healthy or not. Our feature importance is shown on the right. All of the values add up to 1, meaning that each of them are a percent. And according to the table, water has the largest feature importance at 13%, followed by vitamin C, carbohydrates, and then iron.
So our conclusion. So our results should not be the real truth unless someone else confirms our findings. They may be like a hidden factor or Simpson's paradox evident in our research. In which future researchers can research more in detail in order to determine more and learn more about our research. Through manual labeling, we were first told to figure out which of data is healthy or not with a reason explaining. We were then able to find and use a confusion matrix and feature importance showing which parts of our data were important for our project. We had originally tried to use different various methods for finding out whether or not a particular food is healthy using clustering methods. However, we later converted back to manual methods after finding out that the clustering methods were not working as we expected to work, and using manual methods were better since it allowed us to have more accurate data and more accurate research in overall.
Though we didn't finish our research this semester, we do have plans for the future, such as creating more accurate deep learning programs, considering other factors since we mainly focus on sugar, lipids, and cholesterol, considering a time aspect since some foods such as those including sugar are considered less healthy if eaten at different times, adapting our research into a program that can inform the user if the food they're eating is healthy or not, and giving a score to food out of 10 based on how healthy or unhealthy it is.
Acknowledgements. We'd like to thank ASDRP for giving us this opportunity to conduct this research, and as well as Mr. Subramaniam for guiding us throughout this process.
And here are some of the sources that we used.